Okay, we're good. We're good. All right. Welcome back, everybody, to Bio 105. This is day 4. It is Thursday, July 10th. All right. Here we are in the schedule right here. No lab today, right? Just lecture. We're going to try to wrap up that whole PowerPoint, see how we do. Next week is going to be just like this week, except the labs are going to be a little more hardy. You know, there'll be a little more to them. Okay? Third week, all right, you'll have your first lab quiz and your midterm because week 3 is the middle of the summer term. So that's not next week, but it's the end of the following week. Wednesday will be the lab midterm, and then Thursday will be the lecture midterm. Cool? Remember that lab midterm is open notebook. The lecture midterm is not open notebook. Also, don't forget, folks, you know, all these assignments for this week, they're going to be due at 11:59 p.m. on Sunday. So I've seen a lot of people getting through them, making progress. That's awesome. The reading quizzes are probably the lower effort ones for most folks, but they're all important. Complete them all. There's a lot of points there to be gathered. All right? Awesome. Are there any questions before we get back into our PowerPoint on the biological macromolecules? We've only got 43 slides to get through, which is pretty great. It means we're making good progress. At this point last summer, I was behind. So, questions? Let's do it then. Let's get into this. Okay. So, yesterday in lecture, we talked about how these biological macromolecules get synthesized via dehydration synthesis, how they are broken down via hydrolysis, broken from polymers into monomers. And then we started to talk about the biological macromolecules, beginning with carbohydrates. In the carbohydrates, the monomer is the monosaccharide, the polymer is the polysaccharide. Monosaccharide example would be glucose. All right, right here, okay? You join them into a big chain, and they make things like starch or cellulose. Cool? Carbohydrates can play multiple roles. They can be energy storage. That's one primary reason for them. The other one is structural carbohydrates, like cellulose that makes up plant cell walls, okay, and chitin that makes up arthropod exoskeletons. Awesome. That got us into lipids, all right? Lipids, the waxes, the oils, the fats. What's the main thing we know about the lipids that I've said 100,000 times at this point? You're darn right they're hydrophobic. What type of bonds make them hydrophobic? Non-polar covalent bonds, right? Those carbon-hydrogen bonds are non-polar covalent bonds, and the majority of a lipid is hydrocarbon bonds, all right? So we talked about the fatty acids, and we talked about saturated versus unsaturated fatty acids yesterday. Unsaturated fatty acids, okay, have a double bond somewhere in the fatty acid chain. Somewhere there are 2 carbons that are double bound together. And remember, if 2 carbons are bound with a single bond, single covalent bond, there can be rotation, all right? The whole chain can sort of can flop down into a high density flat state, all right? But in a double bond, the molecule can't rotate, and therefore, if you have a cis bond here, all right, or a cis isomer of a fatty acid, there will be a kink in the chain that can't be unbent. And I used the example of saturated fats and trans fats, right? They're like dowels, wooden dowels, and an unsaturated fat with cis bonds is like a branch. You can fit more wood into a barrel if the wood is in the form of dowels than if the wood is in the form of branches. So in other words, these types of fats, these types of fatty acids, can reach higher densities. They can more easily pack into a stable form because there's not this branch, this kink in there, taking up a lot of space. And remember, all of these things are moving, okay? So unless something's at 0 degrees Kelvin, all the molecules in it are wiggling around a little bit. It's just how much are they wiggling around. So the dowel branch analogy in the barrel, also imagine that that whole thing is like shaking at the same time. And you can see how branched fats would not be able to reach as high a density. Make a little bit of sense? Awesome. So, fats, actual fats, not fatty acids. I warned you that they don't really follow our monomer-polymer rules because it's not a long chain of repeating monomers that might be slightly different than one another. Instead, these fats are multiple fatty acids, all right, each one linked to something that's holding them all together. In the case of the fats, it's a glycerol molecule, okay? It gets confusing because, well, yeah, the fatty acid chains are still joined by dehydration synthesis to the glycerol molecule in order to make the triglyceride, okay? However, they are not joined in sequence. Does that sort of make sense? I hope it does. It's okay if it doesn't make a ton of sense. All right. Other things will make sense. You don't have to know everything. It's okay. So 3 fatty acids joined to a glycerol makes a triacyl glycerol, or what we normally refer to as a triglyceride. Sometimes in a lab result, you'll see like triglyceride levels, and it means fat levels. Good. Now, these fatty acids can be saturated or unsaturated, and they can be in the cis or trans isomers if they are unsaturated. All right? So check this out. If you look at these models, you can see that in a saturated fat like butter, it's all dowels, right? It's just a bundle of dowels. So that can easily be packed together. It can reach a higher density. It's more stable. And saturated fats, they tend to be stable and solid at room temperature. Whereas unsaturated fats tend to be liquid at room temperature because they can't all just fall into a stable, solid structure as easily. They're jiggling and they have branches on them, so they're always bumping into each other, okay? So an unsaturated fat with cis bonds, like this one here, will exist at a lower density when it's a bunch of them. And that means that they can be liquid. Remember that rule? We kind of talked about it. Solids are more dense than liquids, and liquids are more dense than gases. And we really only talked about it because water breaks that rule. It all gets confusing. Every rule gets broken by something, right? Does this make sense why an unsaturated fat with a cis bond would exist at liquid? It would exist as a liquid rather than a solid? Make a little bit of sense to folks? Okay, I'm seeing, you know what? I'm seeing a reaction, but it's also Thursday and of our first week, so I'm just gonna put it on that. All right, we'll see some more stuff. Oh, hey, check this out too. Saturated versus unsaturated fats. In your blood, they behave the same, right? In your lymphatic system, they could behave the same. And you don't want a bunch of solid fats in your blood vessels, right? So this is why saturated fats can be considered unhealthy, because they can form plaques. They're hydrophobic, and they can reach higher densities. So it's easier for them to solidify and sort of coat blood vessels and things like that. You don't want that, all right? So people will say, just eat a bunch of olive oil. It's unsaturated fat with cis bonds. It'll help to not form those plaques. And the idea also here is that if you look at a blood vessel that has a plaque on it, right, made up of a hydrophobic substance, like a bunch of saturated fats or trans fats, but we'll get to that, all right? And then you've got some other unsaturated fat particles sort of floating around. Well, they can actually pick up some of this because it's all hydrophobic, and they might be able to take some of it with them, reducing the plaque. There's evidence to support that this can happen. There's evidence to say that maybe this doesn't happen all the time, but it's something to think about. It's all about solubility in water. Your blood's an aqueous solution, right? So these fats aren't very soluble in it, and if they can stick to something, they will. Cool? Which of these is a cis fatty acid? A. Remember, cis, same. Same arrangement on either side of the double bond. Cool? There's a big red circle. Trans fats, all right. Nowadays, it's pretty common. They barely even advertise there's no trans fats on food. But when I was younger, they were removing trans fats from foods, and every snack was advertising that we don't have any trans fats in our food. So you might want to know, like, why are these trans fats so bad? Okay? And what you've learned so far about them is they behave like saturated fats. They can reach higher densities, all right? They're going to be solid at room temperature. They behave like this, okay? So that's the main reason, is a trans fat is basically like eating a saturated fat. But your body sort of has a harder time breaking them down. Those double bonds add additional stability, right? They make the molecule more stable. They make the molecule more shelf stable. So why do we have trans fats if they're unhealthy? Because they're more stable. So this idea of, instead of, you can extract vegetable oils, all right, liquid oils, and then convert them into trans fats and have them be solid and shelf stable for shipping, packaging, and movement. It's hard to move liquids. So the generation of trans fats was really a huge advent. I believe the dude who got credited with it got a Nobel Prize for the whole thing. Question?
Do you have any examples of trans fats?
I don't have one, like a name of one, but they all have names like this. So it's not very exciting, right? I can tell you, though, a little bit about the process of generating the trans fats, which is essentially you take the liquid and you bubble hydrogen gas through it, and that causes double bonds to form. So what you're trying to do is take a unsaturated fat that has cis bonds and convert those into trans bonds so that the substance will solidify. All right. So as far as a name of a good example of a trans fat, I don't have a good one for you. But a lot of processed foods used to contain mini trans fats because they were cheaper. I can't believe it's not butter, right? That's a product. You'll see they'll still market these as like, it's healthy vegetable substitute for butter. Use this instead. But it's just vegetable oil that they have now made into, made it more like a saturated fat. They'll either convert it into a saturated fat or convert it into a trans fat. So you lose all the health benefits of the original oil because it can be processed easily in your body and doesn't reach high levels of density, and converted it into essentially like a butter. That's why it's so hard to believe it isn't butter, because chemically, you're basically eating butter now. But they can say, oh, it's from a plant, so it must be healthy, right? Anyway, trans fats, okay? Hydrogenated vegetable oils, all right? Hydrogenated vegetable oils were a big thing. They're a big ingredient in processed foods. And when you do this, you get saturated fats, and that's kind of what you're going for with like the I can't believe it's not butter, but you also get the trans fats, okay? Then you can extract the two. You can sort of try, there are ways to separate the two with like, I believe you can do it with centrifugation. The details of this process are a little bit fuzzy in my brain. It's been a while. All right, yeah, you got a patent for the process in 1903, making these into saturated fats. So, trans fats, turned out they were leading to heart issues, circulatory issues. They were a great way to transport a source of energy long distances in an era when it wasn't as easy to keep vegetable oils clean and fresh and free of contamination. So there's a benefit for a time, but we don't live in that era anymore. And, now it's hard to get trans fats if you want them. Crisco's got some, right? You can still get it somewhere. It's good, especially if you fry food in it. It's really stable, so you can get it really hot and really fry the food real well. But it's not worth it, right? It's not worth it. Okay, melting points, right? What's a melting point? A melting point is the temperature at which a substance goes from a solid to a liquid. It's really more accurate to think of it as the equilibrium point between solid and liquid, because this is just a little interesting, right? Once you get water to 0 degrees Celsius, it sort of stays at 0 degrees Celsius while it's freezing. The molecules are at the equilibrium point, where they're freezing, liquefying, but the net result is they freeze, okay? So the equilibrium point, if you want to be a stickler about it, for oleic acid, which is an unsaturated fatty acid with a cis bond, is 4 degrees Celsius. That means it becomes a liquid at 4 degrees Celsius. Oleic acid, which is a trans fatty acid right here, has a much higher melting point at 46.5 degrees Celsius. So it'll be solid at room temperature, okay? If you've got more cis bonds, more fluid because there's more branching points. Cool? Awesome. I love it. Phospholipids. I told you they'd be super important, right? Phospholipids are basically just a triglyceride, a fat, except one of the fatty acid chains is replaced with this phosphate group and this choline group right here, okay? Just look here at all of the charges and all the oxygen. There's a nitrogen here, which you know is also pretty electronegative, and there's a positive charge associated with it. This is hydrophilic. You don't have to be a chemist to look at that and say, oh, a bunch of polar molecules could interact with this, because there's regions of charge. So this positively charged region, the oxygen end of water molecules could surround it, and the hydrogen end could surround this negatively charged region. You see? So this head of the phospholipid is hydrophilic, whereas these long hydrocarbon chains here are purely just straight up hydrophobic. This is a molecule with a hydrophilic region and a hydrophobic region. All right? What's the name for that type of molecule? It starts with an A. Amphipathic. Amphipathic. Points. Very good. All right? Amphipathic molecule. That's a good piece of vocab to have locked and loaded. All right? And soap is made up, the active ingredient in a soap is an amphipathic molecule. You're all college students, so you either have watched or you're supposed to watch Fight Club at some point and be like, oh man, I really get it now. I don't know. Maybe there's a new movie that you all watch that does that for you. No, you still just watch Fight Club. That's awesome. Just watch Fight Club. Why not? It's fine. The point of the matter is, remember when they go and they steal all that fat and they turn it into soap. All right? Well, why does that work? Because the fat is a bunch of triglycerides. You run them through a chemical reaction, and you can convert them into amphipathic molecules. And once you've got an amphipathic molecule like this, well, imagine that you've got on your skin, but you don't have to imagine, you are living this nightmare. You are covered in a sheet of oil. And that oil is good for defending you from pathogens and keeping you clean, all right, to an extent. Over time, that oil captures dirt, bacteria start to grow on it, they eat it, it gets bad, all right, you start to smell bad, all right? Soap, okay, is an amphipathic molecule. The hydrophobic region interacts with the oils, and then the hydrophilic region interacts with the water you're putting on your skin. It allows the oils to be pulled off into the solution. That's how soap works, all right? It's an amphipathic molecule. It's pretty cool. In one of your chem courses here, you make soap, or at least you used to make soap, and then you scent it with limonene or limonene, right? You don't do that anymore? No? I used to make soap. It was fun. You made it in a Dixie cup. You took it home. It probably burned somebody's skin because they did it wrong or something and they stopped doing it. Anyways, the point of the matter here is that amphipathic molecules are important in general. All right, one reason being soap. Be careful when you're buying, or be aware when you're buying body washes and things like that. They don't always have soap in them. All right? Sometimes it's just oils and perfumes. And those oils and perfumes or colognes or scents, those don't actually pull the sheet of oil off your body. They just blend with it and fill it full of nice smelling molecules. All right? So I use a liquid soap in the shower personally, but that's just because of a baseline fear of having stagnant oil covering my body for weeks on end. Which I think is normal. We all have that fear, yeah? Okay, cool. Anyway, here's the point, all right? Phospholipids, important amphipathic molecules. They make up the cell membrane. Now, they make up the cell membrane in eukaryotes, they make up the cell membrane in prokaryotes or in bacteria. They don't make up the cell membrane in all of the archaeans, but we don't really talk much about the domain archaea, okay? Solid. We're going to come back to phospholipids, I promise. They're super important. I love talking about them. I already showed you once how if you dump a bunch of these into an aqueous solution, they'll all sort of auto-assemble into what we call a phospholipid bilayer, where all of the hydrophilic heads face out into fluids, aqueous solutions, AQ, and all of the hydrophobic tails face away and make their own sort of hydrophobic domain. That's a phospholipid bilayer. It's what builds the barrier around your nucleus, the barrier around your mitochondria, the barrier around your cells. All right, it's really important. And this hydrophobic solution right here is enough to keep molecules from freely crossing. It makes a semi-permeable membrane. All right. Great. We'll come back to them again for like a third time when we talk cell biology. All right, steroids. You know I know a lot about these. You can tell because of my big muscles that I'm obviously a big juice head. I'm always juicing. I love it, okay? So what are steroids? Steroids are lipids. It's a class of fats, all right? The steroids that people inject are steroid hormones that trigger a change in gene expression, okay, that make you get all beefy, okay? Steroids are a general term because maybe some of you have even been prescribed steroids, and it's like, should I start going to the gym? Well, steroids are a big, broad class molecules, all right? They're lipids themselves, and they're characterized by these carbon skeletons that form rings. But it's still, whenever you see a ring like this, that's just carbon and hydrogen and carbon and hydrogen and carbon and hydrogen. If you look at this structure right here, all of this is completely nonpolar, all right? You have one hydroxyl group on this big molecule. That's a nonpolar molecule, all right? So it's a lipid still. It's a lipid basically made up of a fatty acid that has a ring structure. It's done the thing that the glucose does, where it goes linear to ring to linear to ring, right? We cool with this so far? It's still a lipid. It's a big hydrocarbon. All right? Cool. That's what a steroid is. It's a special type of lipid characterized by four carbon ring structures. Okay? Cholesterol is a steroid. All right? You know about cholesterol. They're always saying you should have a lot of it, you should have a little of it. There's good cholesterol, there's bad cholesterol, LDLs and HDLs. LDLs and HDLs stand for low density and high density lipids. So now that might make a little more sense. It's like, oh, wait, HDLs, high density lipids. I know what a lipid is. I know what density is. All right, is that good or bad? Think it through, all right? So cholesterol is also a steroid, and it is in cell membranes of animals. Why is it in cell membranes of animals? Because the cell membrane of animals has a big hydrophobic region. And there are similar molecules that function like cholesterols that are used by other organisms. What do they do? Well, we'll talk about it, okay? I put some stuff here. I just typed in a little aside. I'll do this occasionally when I'm like, I would love to talk more about this, but I usually end up running out of time. All right? So cholesterol is really interesting. Jamming a bunch of it into a phospholipid bilayer can help to buffer that membrane's fluidity, help to maintain fluidity at an acceptable level. All right? Which is pretty cool. There are other functions as well. Okay. That's good. That's good. Where's my... Oh, membrane fluidity is coming up. I was like, where's my membrane fluidity slide? I was about to freak out. It's coming up when we talk about plasma membranes in general. Question?
Does cholesterol not exist in hydrophilic regions?
It can be there, but it's not going to be soluble, right? So it can be suspended, like in your blood or something. But then if it has a chance to start to settle and conglomerate with additional cholesterol molecules, well, you get exactly the same thing as with the other fats. You get plaques forming. So that's the fear of a high cholesterol diet, is, well, now you got a bunch of these lipids floating around in your blood. They're not soluble in your blood, but they're soluble with one another, so they'll form a block, right? Make sense? Awesome. Very, very good. Okay, cool. That's all the lipid talk. That's all the lipid discussion that we need to do. You've got an idea about, their important characteristics and some examples of lipids. Remember, we've got the oils, fats, waxes, steroids, including cholesterols. All right, cool. And the phospholipids, of course. They're like the types of lipids that we're going to talk about the most in this class. Blah, 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 blah. Proteins. Ready? Here we go. Proteins. The majority of the dry weight of your cell is protein. Of any cell in your body is made up of proteins. Proteins are incredibly important. I've shown you the central dogma. DNA is used as a template for transcription of RNA, which is used as a template for translation, producing a protein. right? So the DNA that you know is really important, all right, is used to direct the synthesis of proteins. The majority of the dry weight of an animal cell, or of most of any cell, if I say any, there'll be an example where it's not the case, but I'm just going to say any. The dry weight of any cell, the majority of it's protein, all right? Proteins are really important, and there are a bunch of different types of them. But their fundamental structure, okay, is consistent. These actually have the monomer-polymer situation going on, right? And we're going to talk about proteins, so we're going to talk about it. All right, here's the central dogma. There's DNA. Look, there's a gene. This is gene 1, okay? There's the sequence of DNA, the sequence of nucleotides that's going to be red. It gets transcribed into the sequence of nucleotides in messenger RNA. Don't worry, we're going to talk a ton about all of this, right? And then that mRNA is read by ribosomes, which it feels like it's been forever since we talked about those. But remember, ribosomes are a subcellular organelle that's found in eukaryotes and prokaryotes. It's not a membrane-bound organelle, because if it was a membrane-bound organelle present in prokaryotes, that would break one of our rules of what makes a prokaryote a prokaryote. They have no membrane-bound organelles, right? So this is carried out by ribosomes, which makes sense. Prokaryotes, eukaryotes, we've all got DNA, okay? If you have DNA, you better have ribosomes. Otherwise, you can't use the DNA to make anything, to make any of the important proteins. Anyway, here's a central dogma. Here's the end product, a protein, all right? We call it a polypeptide because the bond between each amino acid is a peptide bond, okay? That's where the name comes from. Why is it a peptide bond? That's the name that they gave it for linking amino acids. It's all cyclical. Anyways, the monomers, amino acids, tryptophan, phenylalanine, glycine, serine. There's a bunch of them. There's like 23, 24, something like that. We'll talk about that number. I always forget it every single semester to teach this course. All right. They're joined into a sequence, and that big molecule is called a polypeptide or a protein. There are some important types of proteins, all right? Proteins are the most diverse group that we look at here, all right? We talked about the carbohydrates, and yeah, there's a few different things. There's like 2 different things that they can really do. They also are really important as labels on molecules, but we'll talk about that a little later. That's when you jam a fat with you call it like a glycolipid, where you take a fat and you take a carbohydrate and you jam that into the phospholipid bilayer with the carbohydrate sort of sitting out on the outside of your cell, saying like, hello, I'm this type of cell, don't destroy me, all right? Or like, it's where your blood types come from, or carbohydrate labels. But we're not talking carbs. We're talking proteins, all right? Some important protein types. Enzymatic proteins, all right? Enzymes are proteins. What are enzymes? Enzymes are biological catalysts. What do? They encourage certain reactions to happen. They don't really necessarily provide the energy for a reaction to happen, but they can couple an energy-releasing reaction with an energy-requiring reaction, and they can be used to generate high-energy molecules if more energy goes into them than comes out of them. We'll talk about energetics, and that'll make a lot more sense what I mean. But enzymes, what do? They encourage reactions to proceed. We've talked about enzymes a little bit. Remember, lactase, the enzyme for breaking down lactose. They usually have the ASE suffix, and usually the beginning of the name tells you what they break down. Cool? Cool. DNA polymerase is an enzyme that makes a DNA polymer. That's another nice example of it tells you exactly what it does, but that isn't breaking something down, it's making a polymer, it's building something up. And that's going to require energy. But I'm getting way ahead of myself. I just get excited because I know we're going to talk so much about enzymes. For now, you've heard of enzymes before. You know they're in your tum-tum. They help break down food, right, by encouraging certain reactions. Defensive proteins. Antibodies are proteins. Antibodies get released, they latch on to something that's bad, and usually then your body can identify it as bad. All right? And you can develop these antibodies so you can detect the thing again later. Antibodies are proteins. Storage proteins. All right? Storage proteins, they're like an energy storage molecule, but they're not storing energy. So they're nothing like an energy storage molecule. All right? Storage proteins are blobs of protein that have a bunch of them. They're built up of amino acids, and they can easily be broken down to provide the amino acids that you need to build other proteins. So it's just a bucket of Legos, that's all mixed up. Or like, let's say, you actually, I don't know if you did this. This might say a lot about me as a kid. I wanted my Legos to like sit flat in the box. So I clip all the Legos together just to make a long chain of Legos. Anyway, that's essentially like a storage protein. It is a polypeptide. It is a long chain of amino acids, but it itself doesn't do anything. It just exists to be broken down into amino acids. Okay, next, transport proteins. Okay, transport proteins get embedded in the phospholipid bilayer, so they must be hydrophobic to be embedded there in the phospholipid bilayer. Why am I not zooming in? Look at that. That's nice right there. Okay? Transport proteins. They get embedded in the phospholipid bilayer, so they must have a hydrophobic region. Okay? And they allow things to cross that otherwise would not cross the plasma membrane. We're going to talk about that a ton too. We have an entire lecture just on the cell membrane and what sort of stuff goes on there. Okay. Oh, man. That's why I'm not zooming in all the time. It's annoying. We've got one more page, 4 more protein types. Ready? Let's do them. Hormonal proteins. Some hormones are proteins. All right? So a hormone will just, for our purposes, it's a signaling molecule. It elicits some sort of response. It gets released somewhere in your body, like, say, insulin being released in your pancreas or from your pancreas, right? Then it goes through your bloodstream, and when it hits another cell, it makes that cell do something, okay? So insulin is a hormone, okay? It goes into your bloodstream. It makes your body break down glycogen, that storage carbohydrate that we talked about before. All right. Now, cool thing to think about here is because it is a protein, this hormone is a protein, and not all hormones are, just to be aware of that, okay? If you take the gene for the human protein and put it into something else, you can make that other thing produce the human version of that protein. So insulin gets produced by E. coli that's been genetically engineered, right? The human gene for insulin gets taken out, popped into E. coli. You let the E. coli grow, and then you just blend it up and you chug that, and then you're good to go if you have diabetes. That's inaccurate, of course. There's a lot of other steps in getting the insulin out. But it doesn't have to come from like cadavers, like where it used to have to come from, which is pretty nice. Okay. Receptor proteins. A big commonality you're going to see here, right, is that proteins can be embedded in the phospholipid bilayer. We already talked about transport proteins and how they get embedded. Well, there's also these receptor proteins that basically span the length of the phospholipid bilayer, okay? And they can receive a signal from the outside of the cell and then pass that signal to the inside of the cell without ever having to let that molecule cross the bilayer, cross the membrane. So you can say, oh, there's a hormone outside of me. Hey, start doing some work. Start changing genetic transcription patterns. All right? Or, oh my God, that's an enemy. Let's not allow that phospholipid encased thing to come in. There's a lot of reasons for this. So you have receptor proteins. This is an example. This is supposed to be the end of a neuron, right? They're always illustrated like this. And it is releasing neurotransmitters that are then received by a target cell, which are then making that cell do something. Cool. All right. Contractile and motor proteins. All right. Muscles are the reason that muscles can contract is that you have these motor proteins, okay? That basically you zap them with energy and they change their shape. You blast them with some energy and they change their shape. And so if you have two you have these sort of like cables, all right? In this case, it's an actin microfilament. So you've got these two cables, right? And that's your muscle tissue, okay? And then you blast a motor protein that sits in between them with some energy, and it goes chunk, and it pulls them, okay? Like this. And that allows for muscle contractions. And then you just spin these filaments around something, and it can make it pump. Or you have long cables here that then can be shortened by a contraction, and that lets you working with your skeleton, okay, all the anchor points, it allows you to move your body around. All right? All with these motor proteins and these, contractile proteins, it's more of the filaments, all right? They're all these structural proteins that act like cables or tubes and things like that. Actin's an example. Don't worry, we'll talk about them in general. We'll talk about them in more detail when we hit the cell section. Last one, structural proteins. Just like structural carbohydrates, what are they there for? To give structure. Collagen is a structural protein, right? So this tissue that I've got dangling off my ears, all right, it's given structure by all these collagen proteins that make a framework that the cells can sort of exist in. Holds everything together. Also keratin, okay, is a protein. Keratin, like in your hair and in your fingernails, right? So keratin, collagen, those are both proteins. They're structural proteins. The function they carry out is getting structure. Now you'll notice there's no energy storage protein going on here, right? We've seen a storage protein, but it was just to store amino acids. In the example of albumin, oh my gosh, albumin in an egg. Where's that egg? Here we go. Oh, ovalbumin. All right, cool. It's going to be broken down to give amino acids. It's storing amino acids. It's not storing energy. Solid? See the difference there? Your body can break proteins down for energy, but it's not a very good source of energy. But your body is also a jerk, okay? And so it'll be like, yeah, why not? We'll break down some protein for energy. And you're like, solid. I've been trying to eat better. Just eat the fat, body. You stupid body. What are you doing? You know what I mean? Maybe we're having different journeys, okay? Moving on to the building blocks of the protein, the monomer for the polymer that is the protein. The building blocks of a protein are the amino acids, all right? Now here's a chemistry shorthand that you may have seen before. These R groups, all right? Sometimes there'll be a bunch of molecules that are really similar to each other in one region. This is the case with the amino acids. Every amino acid has this, all right? But then there's this side chain, this R group, all right, that's different between each amino acid. So it's nice when you're trying to show these molecules to be like, oh, here's what an amino acid looks like. And then me, as a basic scientist, I can be like, oh, that R group, that indicates to me that there's a bunch of different amino acid types, and each one's different here. See what I'm saying? So all this stuff's the same. And look, functional groups that we've learned already. Here's an amino group right here. Here's a carboxyl group. Remember, it's like a carbonyl group, which would be just C double bound to O, but then it also has a hydroxyl, an OH group bound to it. So, an amino acid is a carbon bound to an amino group and a carboxyl group and a hydrogen, and then something else, something that gives it a specific function. Cool? Okay. I love it. This carbon's called the alpha carbon. You can tell because of its energy. It has alpha carbon energy. Let's look at it. It's red, you see, because it took too many neutronics. Is that a reference? Am I connecting with you all? Is that what they're called? No? They're not neutronics?
Is that like the testosterone thing?
It's like those new brain things that Joe Rogan says you should take. I don't know.
I know what you're talking about.
You know what I'm talking about. I'm old. I'm 36 this month. I might as well be 50. All right. Here are the amino acids. Okay? Look at them. Marvel at their structure. See the little purple part? It's the same in every single one. What's different? The side chain, the R group. See what I mean? I'm going to flip back and then back and see how every single amino acid is the same in the amino group, the carboxyl group, and the alpha carbon, all right? And then how the side chains are different. And you might have a sharp eye and be like, oh, wait a second. This is NH3 plus and this is O minus. It's because that's what they're going to look like when they're in a chain. It's just being shorthanded differently. Don't worry too much about that piece. What you need to know is that they all differ in their side chain. Now, just like I do every semester for the last 14 times I've taught this course or whatever, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20 different amino acids. 20 amino acids. And these 20 amino acids, by being joined in different order, okay, make up all those different types of proteins. There's big variety in those proteins. Do a lot of different things, right? Now, how do we get these proteins to be so varied and so complex and get them to do so many things? Well, it's because of the amount of variety in the amino acids. And the different classes of amino acids can do a bunch of different things, right? For example, for a very simple example, right? There are hydrophobic amino acids and hydrophilic amino acids. So if I want a protein to transport things across a phospholipid bilayer, well, I could build it up of hydrophobic amino acids here and hydrophilic amino acids on both ends, and then it would just go boop and be held in there by hydrophilic interactions. And then imagine that it's a three-dimensional tube, all right? All tubes are three-dimensional. Imagine it's a tube, and the outside of the tube is all hydrophobic and then hydrophilic, but the inside of the tube is all hydrophilic. Well, now you've just made a channel of hydrophilicity where hydrophilic molecules could cross the phospholipid bilayer. And that's how you build an aquaporin, which is a special type of membrane protein to increase rates of water diffusion across a membrane. So polar, non-polar. Also think about this, right? Let's say I've got a long I've got a long polypeptide, right? My arm is now the polypeptide chain. If my hand is hydrophobic and my shoulder is hydrophobic, they're going to do what the phospholipids do. They're going to get beat up and moved around by the water molecules, and eventually they'll be like, oh, thank goodness. We can hang out here together and we'll just both be hydrophobic. And now you've made this little like pocket right here, and maybe that little pocket is full of a bunch of acidic amino acids. Now, if I can get something to go in there, it's in an acidic environment, and it could be broken down more easily. So that's with just the most basic broad strokes. You're already hopefully seeing how, oh, if I built a big chain out of these things, you can make it do a lot of different stuff. All right? The function of each amino acid is dictated by the side chain, which differs between each amino acid. All right? Cool. I just zoom in. Here's the nonpolar ones. Look, they're all nonpolar. Why is this one nonpolar? It has nitrogen. That's very electronegative. It's the shape. Don't worry about it. All right? Cool. Polar side chains, hydrophilic. There they are. These are all pretty straightforward. Electrically charged side chains. All right. Boom, boom, boom. Acidic or basic side chains. Now look at how they marvel at how they bind together, how the bonds are formed through dehydration synthesis. All right? So, the amino acids are joined into a chain in the repeating unit. All right? The repeating piece. Sort of makes sense? The chain isn't formed, none of the side groups, none of the R groups are directly bonded to one another here when you're building up the polymer. To build the polymer, it's all through the interactions between the amino groups and the carboxyl groups. All right? And this forms what we call the backbone of the polypeptide, which is just repeating, right? It's a long repeating identical chain of just the same amino group, carboxyl group, amino group, carboxyl group. Well, it's really amino group, carbon, amino group, carboxyl group, amino group, carbon, carboxyl group, amino group, carbon, carboxyl group. Join, right? So this is all the same. The difference is you've got all these different side groups projecting off of the long repeating backbone. Cool so far? They give these molecules directionality by identifying one end and the other end, but it's pretty straightforward. There's an amino end where the amino group comes off, and then there's a carboxyl end where the carboxyl group comes off. They're called the N-terminus and the C-terminus because one ends with a nitrogen-containing group and one ends with a carbon-based or carbon-containing group. You don't have to memorize that, all right? It's just, I'm just explaining the additional piece here. Cool? Protein structure. Proteins have different levels of structure, okay? At the end of the day, the important protein structure is its three-dimensional structure. That's what gives it its function. The protein's three-dimensional structure is determined by the sequence of amino acids. And remember, if you've got like hydrophobic, they can click together. Or maybe you have like a positive charged here and a negative charged here. So they click together. And then I have a hydrophobic hand and a hydrophobic head, and those fold together, right? So now this long chain starts to make a three-dimensional shape. Make sense? It's all through side chain, well, it's not all through side chain interactions, but the side chain interactions are what are going to give it its three-dimensional structure, really. Let me explain, okay? Usually a functional protein is multiple polypeptides. They get all folded up, all right? And then they associate together. So you make a long polypeptide chain, it gets all twisted and folded up. You make another long polypeptide chain, it gets all twisted and folded up. And then they click together, and that makes them able to do something, right? They go out and they eat up other molecules. There are levels of protein structure. Primary, secondary, tertiary, quaternary, all right? The primary structure is just the sequence of amino acids. Glycine, glycine, serine, tyrosine, glycine, just what amino acids are there in the sequence. The secondary structure is, it results, okay, from interactions between the backbone, okay? So that long chain, that long backbone, okay, check this out. It's got oxygens and nitrogens in it. So it's going to make polar bonds, right? There's going to be a degree of polarity here. And the backbone will interact with itself in a polypeptide and form hydrogen bonds with itself. But because it's repeating, it will make a repeating structure, a really regular structure, like a pleated sheet, which is just like an accordion shape, like this, okay? Just imagine, right? There's hydrogen bonds between the backbone, giving it an accordion shape, okay? Or a alpha helix. It's called the alpha helix and the beta pleated sheet sometimes, all right? The alpha helix, same thing. I can't make my arm twist like that. I can't make a piece of paper fold like that. I'm just not talented enough, okay? But you can imagine that repeating backbone, okay, is forming hydrogen bonds with other portions of the backbone. So this structure is common to most proteins because all the proteins have the same backbone structure, all right? So that pleated sheet, helical structure stuff can happen. Now, how do you get the right backbone interactions to occur in the protein that you're trying to get? It's all outside of the scope of our class, okay? But what you do need to know is that the secondary structure is all due to hydrogen bonds between the backbone and itself, all right? Or hydrogen bonds between different regions of the backbone. Okay. We'll get through tertiary and quaternary structure, then do a lovely 5-minute break, and then finish this up. Tertiary structure. I've already been talking to you about it. I think it's the easiest level of structure to understand because you've seen that there are, for example, positively charged amino acid side chains and negatively charged amino acid side chains. And if I have a positive here and a negative here, they can snap together and give structure to the protein, okay? So the tertiary structure is all due to interactions among the side chains, those R groups. But remember, there's a lot of variety among those R groups. So the types of side chain interactions, there's a lot of variety there as well. And that's what gives each different protein its flavor, its different abilities. All right? Cool. The last one is the quaternary structure, okay? And the quaternary structure is just when multiple fully formed proteins come together. Remember the previous slide? Most proteins are made up of multiple polypeptides that are joined together. So that's the quaternary structure. So let's rapid fire it. Primary structure, sequence of amino acids. Secondary structure, hydrogen bonds in the repeating backbone. Tertiary structure, side chain interactions. Quaternary structure, two fully formed polypeptides with their own primary, secondary, and tertiary structure come together to make an actual functional protein. Cool? I think that, let's see if I have anything else pop up on this slide. Primary structure. All right. Secondary structure, there's the hydrogen bonds. Tertiary structure, here's some side chain interactions. Here's a hydrophobic interaction. Here's an ionic bond formed between a positive and a negative charged one. Here's a hydrogen bond formed, all right? Here's a disulfide bridge, which we're going to pretend we didn't see, okay? As also the side chain interactions. All right? Human growth hormone, there it is, HGH, right? Sick. Quaternary structure, look at this. You got a couple examples here of hemoglobin and collagen. Collagen has three different sort of unique subunit cables that all join together to make this fiber. All right. Here's hemoglobin. It's got alpha and beta subunits that need to be associated together. But each subunit here has its own primary, secondary, and tertiary structure. Okay? Solid. We will come back from our 5-minute break and talk about how a disruption in the primary structure leads to a disruption in the function of a protein in the example of sickle cell anemia. All right? So 5-minute break begins. I'm going to just do a timer on my watch. 5 minutes. Go. Enjoy.